So I'm going to go over mitosis, but let's start with the cell cycle. As you already know, G1 is the primary phase of most cells. They usually stay in G1 for most of their life cycle. Occasionally, though, of course, some of them have to divide, reproduce, and replicate for various reasons, whether it's development, it's reproduction, tissue regeneration, all that kind of stuff. So when that happens, the cells get a signal, and they enter S phase. And S phase is where the DNA is actually copied. So there's twice the amount of DNA, enough to make DNA for the other cell. And then once it's all copied, they go into the G2 phase, which is secondary growth, which means that the cell needs enough energy and nutrients to actually divide into another healthy daughter cell. And so this includes, all kinds of nutrients, all kinds of energy. And then finally, once the cell is ready, it finally goes into mitosis, where it duplicates all the DNA, and so that both cells have full complement. And then cytokinesis, so we'll head on from there. This is a simple pathway from a diploid cell in G phase, and then into S phase where the DNA is actually duplicated enough for two cells. And I don't know. And then finally into G phase, G2, and then into mitosis, which is what we're going to look at. So G2 of interphase, that's the last part of G2, where the cell is getting enough nutrients and everything together is to divide. The centrosomes with the centromerial pairs and the chromatin, all duplicated, all these different things are all set. And then we go into prophase, which is actually the start of mitosis. And in this case, the chromosomes actually start to condense, so you can actually see them. They all have a centromere, which we'll learn about later. The chromosomes are made of two sister chromatids, one for mom, two because we need one for each cell. And then there's these other things that we'll learn about. And then prometaphase, these kinetochores are attached to these microtubules right here. And then they're starting to be pulled apart. And then finally, the pairs of chromosomes are all lined up along the metaphase plate. And then they go into anaphase, being pulled apart into daughter chromosomes. Then there's telophase, where a new nuclear membrane starts to form around each part of the DNA for each nucleus, and then a cleavage furrow follows. So to get into it more exactly, there's these centrosomes with centriole pairs that are going to migrate. The chromatin, of course, has been duplicated. The chromatin is condensed DNA. There's a nucleolus, there's a nuclear envelope, of course, of plasma membrane. Two centrosomes have formed by replication of a single centrosome in animal cells, each with centrosomes featuring two centrioles. Chromosomes duplicated, the light micrograph showing divided lung cell. Okay. Now in prophase, this is the beginning. The chromatin fibers become more tightly coiled. We can actually see them condensing into discrete chromosomes. The nucleoli disappear. Don't worry about that. The mitotic spindle begins to form. It is composed of the centrosomes and the microtubules that extend from them. This is the whole spindle assembly. God damn. Okay. And then the centrosomes start moving away to either one side or the other of this cell. This is the longest stage. The centrosomes are now at opposite ends. And the pairs of chromosomes are now on what's called the metaphase plate. Let's see. Now the chromosome centromeres lie on the metaphase plate. That's right in, let's just see. So the centromeres are right in the middle of these things. These black ones are the kinetochores, and they're actually attached to the spindle fibers. And each one is attached exactly to the kinetochore on one of those chromosomes, and then corresponding, and they're going to be pulled apart. Actually, those are the microtubules, which is the spindle fibers. Okay, and then we go to anaphase, where the chromatids are now pulled apart, and they go to opposite poles. And they're pulled there by the microtubules. And then finally, we have teleophase. So the two daughter nuclei begin to form in the cell. That means there is, let's see, right here. This is going to form a nuclei. The chromosomes become less condensed, and finally there's a cleavage furrow where the two cells are now going to form, or the one cell is going to form two. And then finally you have cytokinesis into separate daughter cells with exactly the same complement of DNA. Also, because we're talking about eukaryotes for the most part, eukaryotes have a whole bunch of organelles, and so they're also transferred, and they're actually transferred as the cell splits, like mitochondria and chloroplasts and things like that. They're also, some go to each side of the cell and then they duplicate there.